We finished our conversation on Thursday with the discussion of the main mechanisms of pathogen recognition, namely pathogen recognition receptors that recognize pathogen-associated molecular patterns. Just as a reminder, you have certain molecular signatures like flagellin or like a peptide that in fact belong to certain class of microorganisms, and then human cells have recognition receptors able to recognize these molecular patterns. Now, these patterns play a really important role not only in recognition, and in the initiation of certain innate responses such as phagocytosis. So this process is non-specific, but when we say it's non-specific, we have to take into account that phagocytes like macrophages and neutrophils still can tell pathogens from our own cells. So they know which cells to destroy and which don't. They really cannot recognize any specific pathogens. Well, neutrophils do not recognize specific pathogens, they just target pathogens in general. Macrophages, it's a little bit more complicated, but we can say that macrophages lack the ability to be specialized to act against a specific pathogen. So, pathogen, like this bacterium, initially binds to the pattern recognition receptors on the phagocytic cell. Then phagocytic cell forms so-called pseudopodia, or false legs, and this pseudopodia engulf a pathogen. Okay, so here you can see the engulfed pathogen in the vesicle that is called a phagosome. The role of the phagosome is basically to transport a pathogen into the phagocyte, and eventually phagosome fuses with a vesicle called lysosome. You remember, lysosomes are intracellular organelles that contain a variety of chemicals such as reactive oxygen species, various proteolytic enzymes capable of destroying proteins and nucleic acids. So when phagosome fuses with lysosome, lysosomal content, these yellow granules, they essentially destroy a pathogen. Whatever is not being destroyed, whatever is a waste, is released from the cell by exocytosis. It should be not phagocytosis, exocytosis. And there are two types of phagocytic cells, as I mentioned before. Two main types for which phagocytosis is the main mode of action. Those are neutrophils, which die in the process of phagocytosis, and macrophages, which survive. Now, I want to kind of, highlight that in fact, the regulation of phagocytic responses is incredibly complex. Just to give you an idea, there are special signals on the cell surface that are called, and that's not my invention, that is a literal name of those signals, eat me signal and don't eat me signal. Seriously, there are special, so there is a molecule, as far as I remember, it's a molecule called CD47, which is a don't eat me signal. When macrophage recognizes CD47 with its receptor, it does not destroy the cell. And you can imagine that tumor cells are pretty good in expressing don't eat me signals. And one of the approaches to the tumor therapy is how to mask or inhibit those don't eat me signals so macrophages will regain the capacity of destroying tumors. That makes sense? Now, another really interesting innate mechanism of immune response to the infection is called a complement. It is a system of plasma proteins. They are not associated with any particular cell that can be activated by three main types of triggers. Now, essentially, the take-home message for you, the idea that I want you to understand is that all the pathways of complement activation, the classical pathway, the lectin pathway, and the alternative pathway, they all converge in this C3b protein in the center. I highlighted it in green. Okay. So what triggers the classical pathway of the complement activation? In classical pathway, antibodies, specifically IgG and IgM, that are bound to the specific pathogen, activate. Another name for it is a fix complement. You can see the antibody-antigen complex here, activating the first protein, complement response C2, C4, C3. Anyway, eventually the classical pathway ends up in activation of C3b protein. Now the lectin pathway, here at the bottom, is activated by a sugar called mannose. So mannose is on the pathogen surface. Mannose binds to another sugar that is called lectin. And now lectin-mannose complex activates the lectin pathway. And you can see that lectin pathway eventually leads to the activation of C3b protein as well. And finally, alternative pathway, shown here on top. In this case, the activation signal are different molecules on the surface of the pathogen, not very specific grouping or anything like that. So basically, the pathogen directly activates C3b. Now, we figured that all three pathways, classical, lectin, and alternative, they all converge on one protein called C3b. So obviously, the C3b protein has multiple effects on the downstream events. So what are the final outcomes of the complement activation? The first one, shown here in the bottom, is the stimulation of inflammatory response. Okay, complement proteins will activate macrophages, will increase phagocytic recruitment, white blood cells will migrate to the site of infection, promoting phagocytosis. Good? Understood? Second big outcome is called opsonization, and I want to stop at this outcome a little bit longer because we will encounter that term again. Opsonization is a binding of, in this case, complement molecules to the surface of, say, bacteria. Now, this sounds very dry, but think about this. It's like complement pours some ketchup on the bacteria, making it more attractive for phagocytes. I mean, we mentioned this idea of Red Queen hypothesis, the competition between pathogens and immune system. So in this case, pathogens, they have a capacity to escape phagocytosis. When complement binds to a bacterial cell, macrophage can phagocytose it more easily. It makes it more prone to phagocytosis. Does that make sense? But the idea here, idea of opsonization, is that complement molecules stick to the surface of the pathogen like a glue. Does that make sense? Like in the department stores, they have this on the clothing. They have this things when you open it, it's going to spray the ink. Kind of, it's kind of, opsonization doesn't kill a pathogen, it just tags it. It just shows macrophage, hey, eat it. Make sense? Okay. And the third outcome is called the membrane attack complex, shown here to the right. Okay. So in membrane attack complex, we have 4 proteins that assemble into a pore that spans through the bacterial or protozoan membrane, essentially disrupting the normal flow across the membrane and leading to the cell lysis. And the pretty common term for membrane attack complex, it's called MAC attack. Okay, so 3 outcomes of the complement activation are stimulation of inflammation and leukocyte recruitment, that's 1 in the bottom. Opsonization, that's 2. And MAC attack. Any questions? Good? Now the last mechanism of innate responses that I wanted to talk about are interferons. You may have heard this term. These molecules are used in clinical practice. So what happens? How are interferons stimulated? Well, we will have to come back to our old friends, pattern recognition receptors. So pattern recognition receptor, such as, say, TLR9. You don't have to write down the specific names, I'm just giving you an example, okay? TLR9 recognizes its corresponding pathogen-associated molecular pattern. For TLR9, it's going to be DNA. And when this recognition happens, TLR9 initiates an intracellular signaling pathway. The proteins activated in that pathway eventually translocate into the nucleus, go into the nucleus, and bind to the promoter for so-called interferon genes. Okay, so these genes get expressed, and interferon is produced by the infected cell. That's important. You see, cell is infected. Make sense? Interferons are not produced in a healthy tissue. Does that make sense? Am I clear? So you've got to have an infection. So there are 2 main types of interferons, very creatively called type 1 and type 2. There are multiple subtypes. You don't need, like, named by Greek letters, you don't need to know that stuff. But an important idea of interferon signaling is that interferons, shown here in red, and these circles too are interferons. So these red interferons bind to their extracellular receptors. Interferons are proteins, therefore water-soluble, therefore cannot cross the membrane, therefore the receptor is on the cell surface. Are we clear? Now, which cells carry interferon receptor? Normally, in a human cell, virtually every cell. Okay? Interestingly enough, look at the table to the left. Interferons produced by this cell, okay, they can act upon that same cell. This is called autocrine signaling. Or they can diffuse a little bit and act on the nearby cell. This is called paracrine signaling. In a patient with an infection, you can detect interferons in the blood, meaning that they act as hormones in the endocrine fashion, triggering the responses in the distant cells. Does that make sense? Now, we talked about how interferons are produced in response to the infection and how they interact with the receptors on the cells, triggering some kind of protection. But what is this protection? Well, it turns out that when interferon binds to the receptor, okay, like this, it initiates a whole cascade of intracellular signals that may lead to, for instance, protein synthesis shutdown. Imagine you're a virus. You come into the cell, you expect ribosomes to be available or protein synthesis enzymes to be available, but they aren't. The whole protein synthesis is shut down, so you can't replicate. There's no replication. Some cells can actually kill themselves, undergo apoptosis. That's pretty common response, actually. That's sort of a side note. In the context of the red queen hypothesis, you will see that when cell is infected, the normal response, sort of optimal response would be to die in a controlled fashion. And if you would look at different viruses, a variety of viruses actually prevent cells from dying too early. Viruses have a capacity to inhibit apoptosis. So cell can survive and maintain virus replication. Does that make sense? So apoptosis. Some interferons can stimulate a variety of genes, so-called interferon-stimulated genes like this ISG15. It's the ribonuclease that can destroy viral RNA. Just, interesting molecule. I had a chance to work with it a little bit. It was absolutely fantastic in inhibiting replication of yellow fever virus. The only problem is that when we tested it on uninfected cells, just to see, what happens if you overexpress that ISG15 gene in just regular cells, cells die in 48 hours. It's like a poison, It kills everything, and it's a question of what it's going to kill first. Is it going to kill the virus first, or is it going to kill the cell first? Somebody's going to die in the end, no matter what. Now, so apoptosis, antiviral responses, such as protein synthesis shutdown or RNA destruction. What else can they do? Well, can they be used in the clinical practice? The answer is yes, they can be. They're used to treat certain types of cancer, and they often are the only choice in treating certain types of infections. For example, for advanced infections such as dengue virus, yellow fever, practically all hemorrhagic fevers, we don't really have any good drugs, so interference is the part of a therapy, not really a good part, but for the lack of a better option. For a long time, interference were used as the main, actually, the only treatment for hepatitis C. The efficiency of that treatment was pretty low. The clearance rate was about 25%. So out of 100 patients that got treated with interferon, only about 25% cleared of virus. We moved we moved very far since then. We now have a fantastic drugs against hepatitis C. Another problem with interferons, they have many side effects. Of these side effects, probably the most notable is clinical depression. It is well known patients on interferon develop various psychological disorders. Questions? Good. So we're moving to adaptive immunity. And we're going to first talk about the differences. So adaptive responses are very specific. Okay? Meaning that adaptive responses are actually targeted against a specific pathogen. Let's say, well, you'll know that. Some of these things, I kind of formalize them for you, but you know that already. You get a vaccine, that's your adaptive immunity that protects you later. If you got chickenpox as a child, chances are you're not going to get any chickenpox later. You're going to get shingles, but that's a different story. Okay? You, God forbid, have mumps as a child, that's your only chance to get mumps. You're not going to get mumps again. Right? Well, there are exquisite exceptions, but general idea that you already have a pretty robust immune response. So they are targeted against particular pathogen, meaning that your mumps response will be ineffective against measles. Makes sense, right? However, to develop these specific responses, your adaptive immune cells require an exposure to a pathogen. So here in the graph you see initial exposure, and it takes quite a bit of time to develop a protective primary response. Okay? So it's slower than innate. You can kind of see it. You have, God forbid, some kind of a cut, and it gets red and inflamed in a matter of minutes. Okay? But in case of, say, vaccination, if you decide to go to South America, you better get yellow fever vaccine. And you should get it not one day before you go to South America. You should get it, there are 2 injections, and you should take them, as far as I remember, at least a month before you visit the country. Does that make sense? However, these responses, the adaptive responses, are more effective. They're systemic. Meaning that cells, effector cells, the adaptive responses enter the circulation, and they can reach virtually every corner of your body. But probably the most notable feature that differentiates adaptive immunity from innate is the ability of adaptive immunity to form immunological memory. Okay. Here on the graph you see that after the secondary exposure, the secondary adaptive immune response is much more robust and occurs much faster compared to T1 and T2. T2 is much slower, meaning that, if you are vaccinated, your immune system will respond to that same pathogen much faster and in much more strong fashion. Okay. So that actually what enables us to, eliminate or protect humanity against multiple infectious diseases. But before we get to the fun stuff, we have to go through the fundamentals and the brief overview of the main cells and main branches. So there are 2 main arms, sometimes they're called arms or branches of adaptive immunity, cellular and humoral. So these names, cellular and humoral, differentiate these 2 branches based on the effectors, based on the mechanisms that used by each arm in response to a pathogen. Cellular branch or cellular arm is basically various types of helper or killer regulatory cells. The humoral branch its actions are mediated by antibodies. Antibodies are proteins. Proteins are water soluble. So humor from Greek means liquid. So humoral means in the liquid, in the fluid. And antibodies are in the plasma, so therefore humoral. Cells that mediate humoral responses are B cells. They are produced and mature in bone marrow. A little detail. So it's kind of logical to assume that B cells got their name because they mature in bone marrow, right? Yeah, logical, B bone marrow. B cells were discovered in chicken. And in chicken they mature in a special organ that is called the bursa of Fabricius. Bursa means like a sac, okay? This is why they got their name. Not because of bone marrow. In chicken they're not in the bone marrow, they're in the bursa of Fabricius. Anyway, when B cells mature in the bone marrow, they seed lymphoid tissue, which is lymph nodes and spleen, and then upon the exposure to the pathogen, they differentiate into plasma cells. And plasma cells produce antibodies. They can target virtually any pathogens you can have antibodies against. Viruses, bacteria, protozoa, fungi, anything. We're clear? Does that make sense? T cells, also generated in the bone marrow, but mature in thymus. Therefore, T cells. No tricky stories here. And then, when they mature in thymus, they migrate to the lymphoid tissue and wait for the pathogen exposure. Once they are exposed, they differentiate into a variety of effector cells. It can be helper cells that regulate the activation of the immune system, produce cytokines, chemokines. Killer or cytotoxic lymphocytes, CD8 cells. As you can guess from the name, cytotoxic lymphocytes kill other cells, infected cells, and cancer cells. Inflammatory Th17 cells, specifically induce inflammation. And T-regulatory cells that actually suppress the immune system and control the intensity and the duration of the innate response to a particular pathogen. Any questions before we move forward? So, since we mentioned that adaptive immune responses are specific, we need to answer the question, what are they specific to? Turns out that microorganisms, from viruses to helminths actually, that infect human body, they contain what's called complete antigens. We usually call them antigens. I want to be very pedantic here and call them complete. I'm not going to use the word complete anymore. When we say antigen, we presume it is complete. Deal? So, two characteristics are necessary for the antigen to be complete. Large and complex molecules. They have to be both large and complex, and you will see that not every large molecule is complex and vice versa. When we say antigen, in 99% cases, we mean a protein of sorts. And that makes perfect sense, because if you think about any kind of pathogen, they secrete an enormous amount of protein products, right? Proteins are what constitutes life. Proteins are the most abundant molecules in cell. So no wonder that main type of antigen is the protein. DNA can serve as an antigen. Carbohydrates can serve as an antigen. Virtually, so those are three main, biggest types of antigens that you can encounter. Proteins being the most important. Now, look at this antigen on the right. So this antigen on the right has three epitopes. Epitope. Again, compound Greek word. Epi means above, topo means place. So it's some elevated area, not really elevated. I mean, it's not literal meaning elevated area. Another name for epitope is antigenic determinant. Now, how I usually describe it when people, when somebody asks me what is antigenic determinant. Your immune system does not recognize the entire protein. Same way, you do not recognize, if you think about it for a second, you do not recognize an entire person. When you know someone, usually you know by, I don't know, hairdo or face, make sense? Or way the person walks. Some very specific characteristics. 1, 2, maybe 3. Does that make sense? So these very specific characteristics, they determine a person for you. In case of an antigen, those antigenic determinant are the parts of an antigen that your immune system recognizes. So basically, immune cells have no business with this gray part. Does that make sense? They do recognize this 3. And how do we know that? You can actually, there's a software that allows you to predict possible epitopes. And you can find out, you can say, okay, I think this is an epitope, this is an antigenic determinant. So here's what I'm going to do. I'm going to change it. I'm going to introduce the mutation into a gene, for instance. I'm going to change this epitope, and I will see if, I don't know, a mouse will still produce an immune response. And if you wonder, can we just take an antigenic determinant like this, like single part of the molecule, and use it to stimulate an immune response? The answer is sometimes we can. Sometimes it works. That makes sense? So, you show somebody a photograph. You don't really, in order to see, do you know this person? You don't have to show the entire body scan, right? You just have to show the photograph. Make sense? So, your immune system produces a unique immune response to each determinant. It is shown in a picture on the left. So you see green antibody is produced to green antigenic determinant, brown to a brown, and yellow to yellow. What it means is that when you get, okay, let me put it in the perspective of the human infection. When you get infected with flu, okay, you don't get a vaccine, say you didn't get a vaccine, you get infected with the flu. Your immune system produces a variety of antibodies that target a bunch of different antigenic determinants found on different proteins of the flu virus. Does that make sense? So you have like a variety, you have a huge number of different kinds of antibodies. Some are more effective, some are less. Does that make sense? That clear? Okay. Do you understand the idea of how, so antigenic determinant on the antigen, on the pathogen, interacts with your immune system? You got it? Now, I told you large and complex. How can large molecule be not complex? Polyethylene. Or, yeah, so this bottle that I hold. Plastic, right? It's high density polyethylene. Polyethylene is molecularly that's the structure. So methylene group, long chain, just this, just long chains of methylenes. That's it. Nothing complex, really. It's like super simple. Does that make sense? So that molecule is enormous size-wise, but it's not complex. So you can, I don't know, put polyethylene material into the human body and the immune system will not even see it. Does that make sense? Good. Now, it cannot be antigens. So we were talking about the immune response, and we mentioned the word antibody. And this is something that you have to remember from now on to explain many immunological events. The antigen, okay, I'm going to put like everything that belongs to a pathogen in the red color here. So the antigen, okay, it's a pathogen part. It is recognized and is bound by antibody that belongs to us, to humans. Is that clear? Makes sense? Okay, cool. So what are antibodies? They are proteins, blood proteins called gamma globulins, and they consist of 4 chains. 2 heavy chains that are identical, and 2 light chains that are identical. So far, we're clear? These chains are held together by disulfide bonds. Okay? And you can see that this molecule has 2 main regions. FAB region, which is variable, and FC region, which is constant. Does that make sense? Now, FAB region, AB, stands for antigen binding site. Okay. So FAB, fragment that binds antigen, antigen binding fragment. Now, FC region is constant. And actually, this part, the FC region, is recognized by the cells. Okay? So in this case, C stands for cells. We're gonna talk about the recognition of FC region by the cells a little bit later. Okay? Just a little bit. So far, we good? Understood? Now, can you tell me why FAB region here is variable and FC region is basically constant? This is not an easy question, but you have to think about what are the functions of these regions. So what is the function of FAB region? Tell me, please.
To bind the antigen.
It binds the antigen. So why does it have to be variable?
Because antigens can be different things.
Many different antigens. Very good, yes. Now, FC region, it can bind cells of our immune system. Basically, that's it. Cells of our immune system. Now, why is it constant?
It has to be constant because the cells of our immune system.
Yeah, we have limited number. You know, we have like T cells, we have some macrophages, and that's it. Make sense? So FAB region has to be very variable to recognize a huge variety of antibodies. FC region is constant because it binds just a few kinds of cells. We clear? Now, that idea about antibodies and antigen makes sense to you? Okay, so we're going to talk about B cell origins and development. So, produced and mature in bone marrow, as we discussed, then they move into lymphoid organs. Now, each cell, and I'm going to go slow, you will stop me if it confuses, like really do not hesitate, say, sorry, it's confusing, can you go over it again? Each B cell on its surface, in its membrane, has a specific kind of B cell receptors. If all folks in this class would represent a B cell, each person is a B cell, each person will have its own kind of B cell receptor. Now, can it be that, two B cells have the same kind? Absolutely. But what I'm trying to tell you is that to take a single B cell, it's going to have only one kind. Does that make sense? Like, say, different types of B cell receptor belong to different colors. Cell can only be green or blue or magenta or maroon. No mix. No rainbows. Got it? Make sense? Good? Okay. So a receptor recognizes a unique antigen. So say this B cell has a receptor that recognizes an M protein of H3N2 influenza virus. Okay? This cell has a B cell receptor that recognizes NS1 protein of yellow fever virus 17D strain. That's it. Nothing more. Just this particular protein. Got it? Now, cells that have the same receptor are called clonal population. They are essentially identical. They're called B cell clone. Is that clear? Now, do you understand the idea of B cell clone? So all B cells in a clone will be able to recognize the same antigen, and only this antigen. Now, before cells are ready to seed the lymphoid organs, they have to go through so-called negative selection. To understand this process, we need to ask ourselves a very simple question. Conceptually, a human protein, any human protein, conceptually, can it be an antigen? Yes. The answer is yes. Not for us, right? Not for us. But say, if you take human protein and inject it into a mouse, for a mouse, it's a foreign object, and it's going to produce antibodies to it. Are we clear? So, what if, during the B cell development, there is a clonal population, there's a clone of B cells that, due to some unfortunate mistake, can recognize and be activated by our own protein, say a myelin protein. Remember myelin in the myelin sheath of neurons? Remember those? Yeah. So imagine you have a population of B cells that can recognize myelin. Okay? So what do you have to do with that type of population? Do you need it? Huh? Say again? Get rid of it. Yeah, this is called negative selection. So cells, B cells, that can recognize and get activated by our own self-antigens, so-called self-reactive B cells, they get destroyed. And if this process fails, what then? Say if the population of B cells that can recognize myelin are released into the wild. Huh? Come on, speak up. You just said it. Just speak it loud. Autoimmune, yeah. Autoimmune disease. In case, if it's myelin, that's multiple sclerosis. Okay, that makes sense? So the maturation of B cells is very tightly controlled. Okay? We're good? So now imagine you have a lymph node with billions and billions of B cells, with thousands, probably millions of different clones. You with me? So say you have 1,000 B cells in the clone. That's very few. Definitely not enough to, by itself, it's not enough to produce any significant response. So what happens when these B cells are exposed to the matching antigen? This is shown here. So this is the bacterial cell. This is a B cell, and those are B cell receptors. Does that make sense? Those B cell receptors recognize the antigen that belongs to that bacterial cell, and this matching of a specific B cell clone that carries a certain receptor and the matching antigen is called clonal selection. So basically it's like, oh yeah, we have Staph aureus coming. This clone, your job is to respond to Staph aureus. Everybody else, walk away. Does that make sense? I mean, there's nobody actually directing this molecular process, but the idea is clear, right? So after the clonal selection, okay, B cells of that selected clone start to proliferate. What does the word proliferation mean? Expand, more, yeah, more, multiply. And they start to differentiate. So they become specific. Okay, so B cell that differentiates, it differentiates into plasma cells. And plasma cells now produce antibodies. Makes sense, right? So this is the whole story. When a certain clone of B cells recognizes the antigen, this clone, and only this clone, starts to expand. So that recognition is clonal selection. Then, when it starts to expand, we call it, no wonder, clonal expansion. And during clonal expansion, B cells differentiate into plasma cells. And plasma cells produce antibodies. And they expand to billions of plasma cells that produce, I don't know, trillions, quadrillions of antibody molecules every day. It's enormous, like a cascade activation. Does that make sense? Now, an important note number one. Here in this picture of B cell receptor interacting with its matching antigen, do you see any T cells? Are there any T cells? No. You know why? Because they're not there. This is called T cell independent activation. Okay? So T cell independent activation. Now, if I made a point out of T cell independent activation, it means there's got to be T cell dependent. There is. We'll chat about it when we'll get to T cells because it's kind of more relevant. Now, sort of take home message from that statement. B cells can recognize the antigen directly. So you take B cells, you take antigen, you mix them together, you get B cell activation, clonal selection, clonal expansion, plasma cells, and antibodies. Then, idea of T cell independent activation. Makes sense? Second question, how many antigens are there? Give me a number. Come on. You can be modest, but you don't have to be modest. What do you think? Huh? Million. It's a good guess. If you consider that there are 5,000 genes in one E. coli, that gives about 5,000 different proteins. How many species of just bacteria are out there? Pff, who knows? So probably Joel's estimation was very conservative, right? We're talking about billions of different potential antigens. Now, each of these potential antigens has got to be recognized by a specific B cell receptor. Is that right? So what, we have billion different kinds of B cell receptors? Well, perhaps so. And what each B cell receptor is encoded, it's a protein, remember, B cell receptor, that thing here, it's a protein. So what, we have billion different genes? Well, human genome counts the most wild estimates 30,000 genes. So how do we fit all that variety in a human genome? The answer is so-called VDJ recombination. Okay? It's like you produce, same way, basically, you produce a limitless number of license plates. Okay? You have 26 letters of the alphabet. You have 10 digits from 0 to 9, and you combine them in a certain way, and the combination's virtually limitless. Does that make sense? So, if you think about the B cell receptor, it has two heavy and two light chains, right? Connected by the bonds. Each chain has constant and variable regions. And the variable regions interact with antigen. So now let's take a look at how the chain is composed. So this specific idea, this specific graph, illustrates the generation of the heavy chain. Okay? So imagine that you have this. Is the fragment of the human genome. Okay? And you have V segments, 1, 2, 3, 4, 5, 6, 7. I just numbered them for the convenience, okay? You have D segments numbered 1 to 8. You have J segments numbered 1 to 4. And you have constant segments numbered 1 to 3. So far, we're good? We clear? So constant segments, we really don't care at this particular point because they're constant, they do not participate in the antigen recognition. Does that make sense? We can forget about them for a second. We clear? Let's focus on V, D, and J. When V stands for variable, D stands for diversity, J stands for joining. I'm not asking what they stand for. Come on. Okay? Now look at this. During the selection process, say number 2 is selected, right? Here, number 5 is selected, and here, the yellow one, number 2, is selected as well. So you have a combination of 2, 5, 2. Okay, that's numbers of the segments, right? Now, if you have 44 variable segments, 27D and 6J, do not memorize the numbers, please. You can imagine that the quantity of the combinations between these segments is huge. You got it? And on top of that, you have multiple different variable and joining segments for the light chain. It was only for heavy. Are we clear? So it's like you have a box of Lego and you try to put it together. Okay? And you use just 3 Lego pieces, but you can use whatever. And the number of these 3 pieces combinations is huge. Makes sense, right? So this produces a staggering 300 billion combinations, of which some are eliminated as self-reactive. That makes sense? Now, one may argue, okay, sure, 300 billion, right? Let's say we will eliminate 1 billion, just like self-reactive, okay? Still have a lot, almost 300 billion to 299. But there are more antigens in the world. Like, is that it? Can we do more? Now, this is not going to be on the exam, but the answer is yes. Turns out, after this, so when your combination is already there, you see combination is there, the gene is kind of produced. This gene undergoes so-called somatic mutations. So there are practically random mutations introduced, so that additionally increases the variability of that FAB, the variable region on the antibody. Yes?
That would take it from 300 billion to just 5%, right?
No idea. Yeah. Yeah. But you know what's funny? You're right. So Jacob's right. It technically takes it to infinite number. The funniest part is that our immune system produces antibodies that are cross-reactive. Meaning that, remember we talked about Campylobacter jejuni, how antibodies against C. jejuni can actually attack your nerve cells and cause Guillain-Barré syndrome. That's cross-reaction. You have antibody that recognizes 2 different, similar, but different antigens. Okay? Or it is a well-known fact in light of a couple years old Zika virus infection that antibodies against virus called dengue and antibodies against Zika virus, they're super similar. They recognize both viruses. So, I mean, sometimes it plays out weirdly. But generally speaking, yes, the number of possibilities is endless. We clear? Now, you don't need to know the numbers, you don't need to know what the letters stand for, but you have to be able to recognize the proper mechanism. Makes sense? Like a description of the mechanism. I explain how we have a variety, and you say, no. It's a combination of genetic fragments, okay, basically. So, see the combination, recombination of genetic fragments, that's it. That's how we produce such an enormous variety. We're good? Antibodies have 5 classes. And I combine the main, so what I'm looking for in terms of your knowledge here. So I'm going to start with the most boring on the right. It's IgD. It's the B cell receptor. Okay. And that's it. Like, seriously, that's it. B cell receptor. That's all I need to know. IgE is another monomeric antibody found in very low quantities. Does not cross placenta or activate complement. Thank God. Its Fc fragment binds to mast cells and basophils. And when it activates them, that leads to allergic response. IgE is the main antibody of the allergy. IgA, really important antibody, found in the basically all secretions, okay, from tears and saliva to mucus and breast milk. In fact, it's the main antibody produced and released in the breast milk that confers the protection during the breastfeeding, protection for a baby. Okay, we good? Immunoglobulin M, pentameric antibody, very important, it can activate complement, and it's the first responder. So it's the first antibody that is produced in response to any infection. Okay? You can actually detect when a person gets an actual infection, the first antibody in the first 6-7 days after the infection, you're going to see elevated levels of IgM. Okay? Fixes complement very well. Finally, the most abundant is immunoglobulin G. So it is the only antibody that can cross placenta, and it can fix complement just as well as IgM does. Now, you see that in IgM, all Fc fragments are stuck together. So IgM does not bind to any cells. Now IgG does. This Fc fragment binds to different phagocytic cells, macrophages or neutrophils. Does that make sense? IgG is the main antibody of any humoral response. When people receive a vaccine, when they do titers, it's basically IgG. Okay? They aren't specifically measuring IgG in clinical practice, but when titer is done, it is the IgG titer, essentially. Is that clear? So, as we mentioned before, that's like a whole story. As we mentioned before, B cells recognize the pathogen using B cell receptor IgD. After the recognition, B cells undergo clonal selection, clonal expansion. They proliferate and differentiate into plasma and memory B cells. Okay, so memory B cells, they just, they form a memory. They don't really kill or, do anything, just form memory. Plasma cells don't kill anything too, they just produce antibodies. Now, here's the profile, look at this. So this is when someone gets exposed to the antigen. Say you get bitten by a mosquito that carries West Nile virus. Okay, after a while, your humoral arm of the response starts to produce IgM. I told you before, primary responders, right? First responders. IgM decline and IgG take over. And in fact, in some clinical cases, if the patient comes in, if you take the blood, like all symptoms, you take the blood at the height of the infection, and you find a considerable level of IgMs, it means that's like, if you take the person in and you take blood and you identify a lot of anti-West Nile IgMs, first infection with West Nile in the life of that person. First time. Does that make sense? And then IgM, they followed by IgGs. And then eventually, if the patient survives, hopefully, titers go down. But after the second response, the level of IgMs is much smaller, but IgGs take over. And they will stay elevated for a much longer period of time. And then if you do the third immunization and the fourth, every time the levels of IgG will go higher and higher. And every time more and more memory B cells will be produced. Does that make sense? And it's important to understand that this response here, the secondary response, is possible because of the primed memory B cells. They can, expand much rapidly. Does that make sense? We're clear about the stages of primary and secondary B cell response. Now, there are several types of humoral immunity based on human involvement and exposure to antigens. So let's talk about human involvement. Natural versus artificial. Does it need any explanation? When you get infected, okay, that's natural. When you get a vaccine, that's artificial. Now, active versus passive. Now, that can be a little bit more tricky. In active, your immune system is actually exposed to antigen. So what are the examples? Like, you actually encounter an antigen. What are the examples of active immunity? Yeah, well, any infection, really any infection, or any V word, vaccine. Now, passive immunity is when antibodies are passively transferred to a patient. Does that make sense? Say here. Okay. That makes sense? So patient here receives the infusion, intravenous infusion of immunoglobulins. For instance, the tetanus shot that you receive after, any possible environmental exposure is the antibodies against tetanus, because they will just, it's fast. Okay, they're in the system, they will stop the microorganism. Antivenin, the drug that blocks the action of the snake venom. Antibodies, the passive artificial immunity. Does that make sense? So it's passive. You just receive antibodies, and it's artificial because somebody else injects them to you. Now, what is natural passive immunity? Breastfeeding. Almost typical. Okay. That makes sense. So on the test, that's pretty simple. You will have to distinguish, natural versus artificial, passive versus active. I give you an example and you pick the right characteristic. Good? Clear? Now, what can antibodies do? What are the actions? So first, like really big statement. Antibodies never lies or kill anything. They tag. Am I clear? Remember, 40 minutes ago we were talking about complement. And we mentioned that complement doesn't kill, well, that opsonization thing, when complement proteins bind to a pathogen, they don't kill it. They just tag it. So antibodies tag the pathogen as well, but they're more specific. We're clear so far. So, and it turns out that antibodies, just like complement proteins, can opsonize a pathogen, making it more attractive for phagocytes, therefore increasing phagocytosis. Is that good? Okay. What else? They can precipitate. Here you can see antibodies that bind diphtheria toxin. So when you receive DTaP, the result of that vaccination, your immune system produces antibodies against the diphtheria toxin. When, God forbid, you get exposed to diphtheria, these antibodies block the action of the toxin by binding it and forming an immune complex. This is called a precipitation, bless you. And these immune complexes, these precipitated antigens, can then be what? Phagocytosed. Now, in case of the viruses, the characteristic antibody response is neutralization. So this is virus. Remind me, please, what does virus need to attach to the cell that it's about to infect? Receptor. Now look at this poor virus over here. It's all surrounded by antibodies. can it bind to a receptor now? No, it can't. It's all like blocked, right? The entire surface of the virus is blocked. So this virus actually neutralizing antibodies, they do not eliminate the virus from your bloodstream or from your system. They just prevent the virus from infecting your cells. Does it make sense? Okay? And then viruses can agglutinate. IgMs are fantastic at that. So here you see bacterial cells being clumped together by IgMs. Now, that clumping, that agglutination, first, prevents the spread, and second, makes this enormous chunks of agglutinated bacteria, again, more attractive for phagocytes. And as you remember, IgMs and IgGs can activate a complement. And there you go. Complement activation leads to killing of bacterial cells. So in a nutshell, antibodies do not kill, do not tag, do not kill or lyse, they tag pathogen. They may neutralize it if it's a virus. They may precipitate it if it's a small antigen like a toxin. They may opsonize bacteria, making it more accessible for phagocytes to kill. They may agglutinate the bacteria, preventing their spread. And IgGs and IgMs can then activate complement, therefore leading to MAC attack or increased inflammation, leukocyte recruitment, so on and so forth. Does that make sense? Again, match the description of the action with the name. We're going to take a break.